Students, welcome to my screencast on COVID-19. I miss you. Wish that we could be together, but we are doing our part for physical distancing to prevent the spread of this virus. So this kind of grew out of your COVID project. And so what am I going to do? I'm going to go over some vocab around this viral pandemic. I'm going to go over what is a virus, what's going on with this coronavirus. Here's my model of one that you can make as well. I posted in Google Classroom. We'll do a little bit on evolution, how it is transmitted, and then some help at the end. How can you help? What's going on in the world to do that? Just like all of my videos, this is not for profit. It's only meant to help students. There can be no commercial gain from it. The images I get from Khan Academy and OpenStax, you can access them on khanacademy.org and openstax.org. And this is out of Rice University. And I would also like to credit Gretel von Bargen, an IB biology teacher in Seattle, who put together a really prolific slideshow on COVID-19 that a lot of teachers are looking at and commenting on. And I got some of the ideas for this and pictures from that as well. Okay, pause the video. What's the vocab? What do these words mean? Unpause. Okay, hopefully you got something that the coronavirus is like a branch in one of these trees, right? Doesn't this look like this could be up in Jebel Akhdar? And so here would be a branch, and there'd be these different tips that come off of it, and those would be the viruses. And here they are: OC43, HKU1, NL63, MERS, SARS, and SARS-CoV-2. These are the 7 coronaviruses that can infect humans. These first 4 just give you the common cold. And you probably had one of them before and you've defeated it, right? And then this one can give you MERS, SARS, and then this one's like SARS, but different in that it gives you COVID-19. COVID-19 is the disease when this SARS-CoV-2 virus infects your airways, infects your lungs, and causes what's called a cytokine storm, or a big problems with your immune system in your lungs. And there's differing severities or reactions to getting this virus for different people. What is a virus? So pause the video, see if you can talk that out with a friend. Unpause. Hopefully you got down that it's an obligate intracellular parasite. Actually, hopefully you didn't, but that's what I'm going to tell you. So let's figure out what this word is, right? What does it mean to be obligated to do something? Well, it means that you have to, right? So you've got to do your homework. You got to do your chores. I got to pay taxes, do other stuff. Make sure I'm home at a certain time. I am obligated. So the virus is an obligated thing. Intra means within, and cellular means the cell. So it has to be within the cell, and it's a parasite. Okay, so that makes more sense. So in other words, viruses can't exist without cells. There are more viruses on Earth than there are stars in the universe. That's amazing. There's more viruses, in the country of Oman than there are grains of sand on the Oman beaches and in the Hiba Desert. Okay, so there's lots of viruses. I thought that this might be helpful just to see some scale. Here is a bacteriophage. This is a virus that infects bacterium. And here is the actual bacteria. So this would be an E. coli. Here's the E. coli, and here's a regular colon cell. Look at how much smaller the bacteria is than the animal cell, than the eukaryotic cell right here. And notice how much smaller is the viruses than just the bacterium. This is a liter of seawater that's been stained just to reveal all the different viruses that are in it. So before you get freaked out and you're like, oh, there's viruses in the air around me, which there are, right? Those viruses aren't hurting you. They're not messing with you and your body, right? There's viruses inside of you. It's called the human virium, but they're not hurting you negatively right now. It's just the bad ones that get all the rap. And so this is pretty cool. It's showing that 95 to 98% of the living weight in oceans is microscopic. When we go out in the oceans, and I'm guilty of this, I go out on my friends' boats and I think, wow, I hope I catch tuna today, or I see a whale or a dolphin. And I think if I were to add up the mass of everything in the ocean, that these things would be the biggest and that they would take up the most weight. But in reality, there's so many more bacteria, phytoplankton, viruses in the ocean that they make up 95% of all the weight of everything in the ocean. So think about that the next time you go out on a boat. that there's all this stuff out there just waiting to be looked at, discovered, analyzed, and experimented with scientifically. So the world of microbiology is happening. Okay, here is a picture of a virus life cycle from Khan Academy. And so let's once again think about obligate intracellular parasite. So let me get my pointer. So here the virus is going to come and attach to a receptor on the cell, just like a protein-based hormone, right? And so it's going to attach there, and then the cell is going to say, hello, you have the right key, come on in. So if the S spike on this coronavirus matches up with the ACE2 receptor on one of my respiratory cells, the cell brings it on in. Then the virus capsid, that means the protein coating of it's going to come apart. The inside of the cell often has a different pH, so it's going to change the conformational shape of that protein. Then the viral genetic code gets out. It could be RNA, DNA, etc. But regardless, it can use the stuff within our cells. It'd be like if I went into your house and cooked a meal using all of your stuff. I brought my instructions, right? My recipe, my genetic code, but I used your pots and pans and I used your food that you went and got from the grocery store, etc. That's what this virus is doing here, right? All it's given is this viral RNA, and it's being read by things called ribosomes. And those ribosomes are then making more viral proteins. They're getting assembled into more viruses, and eventually the virus will leave the cell. It can either cause the cell to explode in what's called lysis, or it can jump out in little budding parts. All right, let's talk a little bit about viral evolution. I'm going to take a quick water break first. All right, back. So here is a phylogenetic tree. Think of all the evolution that led to you. All right, we started out on primordial Earth as some cells, and those cells came together and they made colonies, and then they made different ocean life, and then life came out of the ocean, and it made amphibians and reptiles, and then the dinosaurs came about, and then the rise of mammals, and it led to you, right? And that's a very human-centric view of evolution, right? I prefer to tell my students that humans are not the end product of evolution, right? Just because humans are here, this was Darwin's famous phylogenetic tree, by the way. Maybe bacteria over here. Bacteria over here long before humans, and they'll be here long after humans are gone. So it doesn't mean that we are the end product, but just think of all the stuff that led to you. Viruses have been very successful evolutionarily, but they've done the opposite approach, right? They went for a minimalist approach. They're very small. They're ubiquitous. They're everywhere. They just use proteins and genetic material, and they don't have anything else, right? They don't have a heart or the liver or these different organs that you have that you coordinate to be the wonderful person you are. So what are some theories of how viruses evolve? There's a theory that they're just parasites on larger cells that have kind of existed, that they evolve from bits of DNA or RNA that escape from cells, right? When bacterium died, they're able to release their DNA into their environment. So maybe those came together in some catalytic type way to become a virus. Or maybe viruses were around before the LUCA, the last universal common ancestor, and have been around since then, and just been going back and forth infecting bacterium and other life forms. And then the bacterium and the other life forms evolve ways to defeat the viruses, and they just keep going on in this evolutionary arms race. That's what's led to CRISPR, right? This genetic engineering tool where we can cut DNA. We got that from bacteria. Same thing with restriction enzymes. Bacteria evolved those to deal with viruses constantly infecting them. Regardless of their origin, viruses cause evolution. So what do you think of evolution? Now I've gone over it a little bit. And if you take my class, you know that evolution is change over time or a change in the gene pool, right? And so viruses help to cause that. They help to promote genetic diversity in other organisms, and that's because they can integrate their genetic information into them. It's actually seen in humans who are pregnant when they release the hemo protein. We know that came from a virus. So there's been some babies here at TASM lately, and so part of the proteins that were released in that were from viruses that infected our ancestors millions of years ago, right? And think about it, you're like, well, how could that be passed down? Well, if the virus infects what's called a germline cell, which could then develop into a sperm or an egg, then it'd be passed down into the offspring. About 8% of our DNA comes from viruses, is estimated. So that's pretty crazy, right? And E.O. Wilson is a famous biologist who's done a lot of work with biodiversity. And he said that the variety of genes on the planet in viruses exceeds or is likely to exceed that in all the rest of life combined. Pretty amazing. Okay, so here is some of the diversity of viruses, right? We're going to focus on this one, the coronavirus. Here's what Ebola looks like, herpes, rabies, smallpox, different ones, right? When your parents were in school, they might have learned to classify them based on shape or genetic structure or what diseases they called. But now we mostly classify them on how they make RNA. We'll go over that in the next slide, what RNA does. This is called the Baltimore classification system, named after a scientist, not the Charm City. So here is what we're talking about. All these different types of viruses are all trying to make RNA to then make proteins, right? Because that's all they're doing is making RNA, and then it's going to use the cell's own machinery to make the proteins. And just so you know, the coronavirus that we're studying is in group 4 here. It's able to essentially dump its genetic material, this RNA, the blue stuff here, into the cell, and you can start reading. So let's go over what RNA is and all that stuff really quickly, because I've just been kind of spewing those words out. It's my lovely wife, or this could be me in a classroom, right? We're both teachers here at TASA. So as teachers, we're not supposed to leave the class during class, right? We need to be there. We would be like the DNA. So we are inside the nucleus, right? Or you can think it's something that can't leave, right? And you might have learned that DNA is made of 4 letters, A's, T's, C's, and G's. A section of DNA, or a bunch of these letters together, is what is called a gene. And genes code for proteins, right? And you have about 30,000 genes inside of you that you got. You get one set of them from mom and one set of them from your father, and they're going to code for different proteins, which makes you the wonderful person that you are. So she can't leave the classroom. So she's going to send her kids off to go do something, right? This is how one of the interview questions I had to get this job was describing the central dogma. And so here she's going to send out our kids, Johnny, Ellie, and Rory, to go do something. And they're going to be the RNA, right? Another way you can think about it is if you wanted to make a recipe. Here's the cookbook. The cookbook is the DNA. The RNA is going to just be a copy of that recipe that you take to the store. It is single-stranded. It can fit through the pores in the nucleus and go off and do stuff. It uses the same bases minus the T. The T becomes a U in RNA. All right, you can remember that because RNA loves you. It's then going to go off and do something. It's going to make the product, right? And so for here, our kids, maybe we decided to go ask them to get us a Diet Coke. We needed a little caffeine as we make videos and do stuff for virtual school. And so they go and assemble the protein or go get the protein. So let's think about this in the form of insulin. You just ate a bunch of donuts. You had bought the donuts to last you during this physical distancing time, but you ate all of them at once. So then your body has a lot of sugar in it. Your body says, hey, we need to get these sugar levels down. So we're going to access the insulin gene, which is the DNA, and we're going to do this in the pancreas. It's going to send a message out into its cells that is RNA, and then that RNA is going to be made into, it's going to be read at a ribosome and become the insulin protein. And so that's what viruses are doing, right? They're just inserting their genetic information and they're getting proteins made. This works because these letters are the same and they work the same inside of you, inside of your mom, inside of your dog, inside of your cat, inside of your pet fish. Everything uses the same genetic code. It is a universal genetic code. And so therefore viruses can use the same thing, right? And so if you were to put a strand from a tree, like a tree RNA inside of you, you could make whatever protein it wanted to make, right? It might not work. It's not like you can all of a sudden become photosynthetic, but your cells could read it, is what I'm trying to say. All right, so we need to get this back to the coronavirus. All right, and so you know that things evolve, that gene pools change over time. Populations evolve, not individuals, right? One virus on its own cannot evolve, but them together or in a group, they can change the entire gene pool and evolve. Just like you don't evolve, you were born the way you are, and you now you might work out and become healthier, which you should, but then you can pass on your traits. Regardless, here's an example of recombination. So the human strain, the bird strain, they come together and make this bird flu strain that is bad. Here's an example that I just came up with, or that I used in class. The fat cat ate the rat. So your body reads DNA in sets of 3. So for every 3 letters, it codes for a specific amino acid. There's 20 amino acids. You get them from the food you eat, and your body naturally synthesizes some of them. So here in this sentence, there's going to be a mutation. The F is deleted. It changes the whole sentence. Here, the fat cat ate the bat. Well, that's subliminal, right? Because we're talking about coronavirus and bats. So the rat was changed to a B. So it changed the meaning of the sentence. Here, the fat cat, the rat. Well, here I inserted another A. So it's going to change the meaning of the sentence. What I'm trying to show you here is that when viruses or when our cells mutate, right, and every time, like when you have a child, you're going to pass down inherent mutations. They can change the meaning of the protein, and those can be selected for. And so the coronavirus is an RNA-based genome, and RNA mutates at a higher rate than DNA because it's single-stranded and it doesn't have the same proofreading mechanisms. So let's look over here, and we're going to see some different viruses that I've made up, these different colors, and they're going to evolve, and eventually one of them might evolve the ability to escape out of this bat and to infect humans, right? And that's what happened with SARS-CoV-2 in Wuhan. It's not that it wants to. Evolution is not goal-oriented. It's just taking advantage of different changes in the environment, and when there's a change in the environment that selects for that strain of virus, it will keep on getting selected for. So here are our colors. They're all starting to reproduce. We'll say that the red one dies off. This would be the first time I made this video, which I scratched. I was talking about how I wanted to score a goal in Saturday soccer eventually, right? So these viruses are here. I just thought it'd be nice with a picture of our field in there. So here you can start to see a recombination event between the yellow and green strain of viruses. And that's going to make this hybrid. This recombination virus is going to be successful, and it's going to keep on going on. Over here, this gray line mutated and became this black and pink ball. When I looked up black and gray ball, I couldn't get anything. All right, and so this is the one, this one mutated, these kept reproducing, etc. But this is the one that gets out of the bats. And in the wet market in Wuhan, there's some theories that it came from pangolins, but we're going to show you with some DNA sequencing that it's most likely bats. But it doesn't mean that it couldn't have gone from bats to pangolins and then to us. One thing that's important to remember here is that mutation is random. It's not like the virus says, I want to mutate. It just happens. But then natural selection is not. Natural selection sorts out the winners and losers depending upon the environment that is there, right? The mutation makes these random things happen, these random differences in the genetic code that can be expressed as proteins, or it could have been a recombination event between 2 strains of virus. This works in humans too, right? Mutation and sex brings together different combinations of genes that can be selected for, right? So after the spillover event, so after it came out of bats and went to humans, most of it, there's a good stated clearly video on this to check out with John Perry. But sometimes they'll just die off. But if it can propagate or keep going on in humans, then it's going to keep selecting for the virus that's best able to invade human cells, replicate, and move on. All right. Here is a phylogenetic tree of all the different types of coronaviruses. Notice how there's a lot more. We just tend to think in human-centric terms about the 7 that can infect us, but there's a lot more. And scientists will study these, and they're looking at these as they're trying to study COVID-19. And here's just the animal reservoirs that it goes back and forth with. Let's get back to and look at our virus, SARS-CoV-2. Remember that that's the name of the virus that causes the disease COVID-19. Here it is. It's an RNA-based virus. The RNA genome is going to be coiled on the inside. It's going to have a small capsid around it, not really featured here. And then this envelope is interesting because it's going to be comprised of some proteins that were made by the viral RNA inside the cell and by the phospholipid membrane of the cell as it leaves, right? So it's using some of the outer parts of the cell. Let's see how this one's taking it off. It takes it and says, hey, I'm going to go with this. So an example might be if I, went to your house and I got inside because I had a key that was similar to your key, and then I took like your shirt, or I took a costume you had and I went off with it, and I could maybe impersonate you. And so that's what's going on here. There are a couple of important parts of this virus that we need to look at. These are the spikes that are going to go off. And so the M and the E, here's the M protein and the E protein. They're all transmembrane proteins. That means they go from the inside to the outside. And the M and the E work together with the virus assembly. They're going to say, okay, we've gotten inside of John Gibney's respiratory cell, and we're going to start spreading. This S1 is the big problem. The S1, the orange one on my model right here, is going to dock with what's called an ACE2 receptor on your cells. Same one that SARS did. And when it docks there, it's going to be able to trick the cell into letting it in. All right, and so let's look a little bit about the genome of it. This would be the RNA. This is simplified, and it's got different parts of it that code for different proteins. Here it's going to code for an enzyme called replicase. Replicase is going to help to copy the viral DNA. And then here it's coding for the S, the E, the M, and the N spikes. If you've taken some advanced biology, you'll notice that this looks just like mRNA in a eukaryotic cell or a human cell, right? It's got a cap to protect it, and it's got a long A tail that helps to protect it from enzymes that chop up RNA viruses. So it's got all that going on in there. And here is it, once it's inside, it can start to make these things, right? And then it's going to get the phospholipid outer coating by escaping through the membrane. So the transmission, here it is. Here's our coronavirus. It's going to attach to that ACE2 receptor. Well, it's not like the ACE2 receptor was made to get coronavirus. Like, couldn't we just inhibit all these ACE2 receptors, just block them all? That would get rid of the virus, right? Well, the problem is that ACE2 is involved in blood pressure. It's really important. And so the virus enters through your nose, mouth, and eyes. And so it can get in there, usually from an air droplet, right? Or from your hands touching something, which is why it's so important to wash your hands with soap. It can then attach to cells in your airways. As some of the upper cells start dying, as the virus kills them, it can get lower and lower into your respiratory tract and cause more problems. So it's going to enter the cell. It's going to engage in biosynthesis. Think about what that word means, making more copies of itself, of the viral molecules. And then it's going to bud and release. And once again, it's going to use your stuff, like it's using your Golgi apparatus, your endoplasmic reticulum, to make these different proteins for it. Okay, oops. So the receptors are specific, right? So the only door that I can open at school, because I'm a teacher, I don't have a master key or anything, is room 229. Well, I guess in the science closet, right? But I can't open Miss Knudsen's room, Miss Kelly's room, or Mr. Webster's room, right? These are all in the hallway with me. I don't have the key to them. And I don't want the key to them, right? I don't want to be responsible for their rooms. So the same thing happens for the coronavirus. The only thing that it can dock with is that ACE2 receptor. So ACE2 is in lots of cells in the body. So there's some evidence that it can infect more than just your lungs, right? And that would be because your liver and other cells also have ACE2. It's going to infect the lungs first because that's where it's docking and is how it's getting into your body. And then I already said this, that it deals with blood pressure. So it's not like we can just block up all of your ACE2s. You might think, well, why is this called ACE2 and not ACE? There is an ACE. And ACE, you might have heard of ACE inhibitors, and it also deals with blood pressure. And so what's interesting is that it's going to dock at this ACE2, but that's not it. It also needs another protein, another enzyme called furin that your cell is going to release. And your cell is going to release that because it thinks that it's dealing with something with blood pressure. And that furin is going to go to the S spike. It's going to cleave and it's going to cut it in half so that way it can enter the cell. Here is what the ACE2 receptor looks like. The reason I put this slide in here is just so that you can see it's not as simple as like a little green stick as represented here, right? This would be a model. It's a lot more complicated with different amino acids and how it looks, and that, the structure of at least this function. It's a big key in biology. Okay, I wrote right here, cue the NCBI, the National Center for Biotechnology Information. Here is the first coronavirus, SARS-CoV-2 virus sequence from Wuhan. And so this was an early patient who went to the hospital complaining of lung problems, and they sequenced the the RNA from this virus and posted it online. And so they've been doing that for a bunch of them, and you can see how it changes over time. So here it is. This is a completely free website. I just want to show you these are all stand for different amino acids. When prom proposals were cool, I told students to spell out, will you go to prom with me in amino acids with the genetic code, but it became really long and nobody took me up on it. Here is, let's find gene S. And so I had it earlier. Well, I'm going to pause for a second. The old Control F works like a charm. So this is the gene for this glycoprotein, or the spike, that's going to dock with those ACE2 receptors. So I'm going to click on it right here. And this is going to give me the code for it at some point down here. Well, it will. I'm going to click run blast. Let me just find it real quick. Here it is. It's not going to work right now. Okay, I'm going to pause for a second. Okay, now we're up and running. So we're going to blast that spike, the S spike, from the virus. All right, and what this is doing as it's running this search is it's looking through all the different genomes that are publicly available and seeing what matches to it. It'd be like looking through every paragraph in every book in the high school library at TASM and figuring out which sentences match the most with what we have here. Right? And so it takes a little while for it to run. We're gonna let it run, and I'm gonna pause it till it's done running. We're back. All right, so it ran our blast, and you'll see that it comes up, and the highest hit is just, these are different human people that have been sequenced for coronavirus, for SARS-CoV-2, and then you'll see the first organism that it hits is a bat. And so here it is, it hits 100% on that. So it's coming as a crossover from that. So that's how we kind of got that information. I thought you might find that cool. All right, let's go back to the slides. We're almost done. All right, so understanding the structure of this is important because then we can help to try and prevent infection. So we've gone through, there's a game called Folded. It's actually closed for now, but it's a protein folding game where they were trying to figure out the different proteins here and how they folded, et cetera. And so it's actually video game people helping to figure out if we could make an inhibitor against it. Also, Having this stuff open sequence is really important, because as soon as somebody's infected, if they sequence the genome of the coronavirus and infected them, we can try and see where it came from. And because think about it this way, once the mutation happens, all of the subsequent viruses from that mutation will have that mutation, right? So if I all of a sudden have, if my child has a mutation for green hair, then all of his children would have that mutation for green hair, because we're taking sex out of it, right? These are reproducing asexually, right? And so you can see the mutations from them, and you can see, well, was this person, did they get the virus from Wuhan, or did they get it from somewhere close by here? And then you can start to see how it's spreading. So I just wanted to show the tab for this. Here was the folded game, but it's closed. You can follow them on Twitter. It's kind of cool. Here is the next strain site. So it's kind of showing the origination in Wuhan, and then the spread. And then there's Italy, and then there's been the spread to the US. And so it's pretty interesting to look at the cases near Seattle, and you could see that it was pretty close to the original Wuhan strain, and that had probably been being passed around there for quite a while before they put the quarantines in effect. Here is Oman. So we don't have any sequence cases here, but we know that most of the COVID-19 in our country has come from Iran, most likely. All right. And so, oh no. All right, we got to finish up. We're getting long-winded here. And so what are some ways to prevent this, right? Well, soap would help to break apart that that outer membrane that came from your cell, right? And it's because soap's able to grab onto parts of it and pull it apart. So soap's better than even alcohol on your hands, right? And so you should use that. You should wash your hands to the tune of Happy Birthday so that it's long enough. By now, you know all about physical distancing, right? And the point about flattening the curve. And this is really important. And the point of it is, right, is to not overwhelm our healthcare system. So that way we can try and treat this. You might say to yourself, most of the students here, you're young and you're healthy, but you don't know which of your classmates might be predisposed to a disease or have, heart complications, et cetera. Things like obesity and having heart complications are really high up there on getting super sick from COVID-19. So, like my dad has had heart issues before, so he definitely does not want to get COVID-19. So the solutions would be physical distancing so we don't overwhelm our healthcare system. And then as of right now, all we can really do is provide supportive care. And that's what you can usually just do for a virus, giving them fluids and oxygen, et cetera. There are some antiviral drugs, but we don't have any right now that can stop or deal with COVID as of this recording on March 26th. What's the goal? The goal is to get a vaccine, right? And a vaccine is what helps to prime your body so that you don't get a disease like COVID-19 or get as sick as that. There's currently work on one, and I believe a company called Moderna out in Seattle, and there's a lot of work going on all over the place. But it's going to take a while to get that vaccine to trial and to get it approved for human use. So we really need to engage in this physical distancing. There's some good work going on to see if currently available medications could help to stop the virus from going on. So could some other drugs that have been to treat different viruses, could they work on this, right? But that would need to be done in an experiment, in a scientific setting. It's not something that you should just willy-nilly do at home. Could we develop some drugs to block the viral mRNA now that we've sequenced the genes on the inside? And in some hospitals are trying antibody transfusions, right? People who have survived coronavirus, could we give them their blood, the survivors, to people who are infected with it? And maybe the antibodies in there will help to combat the virus, right? And it sounds good, but you need to get your body making its own antibodies, which is what a vaccine does. So how do vaccines work? And this is my last slide, right? An inactive form of the disease is put in the body. The body creates antibodies to defend itself, and they go around in what's called memory cells, right? And so You don't know what polio is and smallpox and some of these diseases because they've been eradicated because of vaccines. And so hopefully the goal would be that we can make a vaccine that would prime your body to have these memory cells to do it. You can think about your immune system as kind of like two things trying to fight off a disease, right? First of all, there's your innate immune system. It's like your skin and stuff that's just going to protect you. And then there's your adaptive immune system that we have as vertebrates. And in your adaptive immune system, there are T cells and B cells. And the killer T cells are going to go around and try and kill things that are non-self, that aren't good, that don't have my print on it as John Gibney. The B cells are going to hang out in your lymph nodes. This is why your neck gets swollen when you're sick. And in your lymph node, those B cells are going to say, hey, here is an antigen. It's going to be presented to it by an antigen-presenting cell. And it's going to say, okay, let me search my antibody database. And what's really cool for your B cells is that they have some enzymes that jumble up their DNA, right? And it's going to jumble up the DNA to create essentially an infinite variety of antibodies. So the antibodies are out there to fight the coronavirus. The question is, can your body make them fast enough that you don't get super sick in your lungs? And so a vaccine would make that work, right? Because then you would have these memory cells floating around, and instead of taking a week or longer to search for the correct antibody in your lymph nodes, it'd be ready instantly. And I'll leave you with this quote, right? Years later, when Jonas Salk, who got this from working with HeLa cells, so thank you, Henrietta Lacks, was asked who owned the patent to his polio vaccine. He said, the people, right? There is no patent. Could you patent the sun? And so I hope that that comes out of this too. All right, students, I'm looking forward to your work on coronavirus. And I'm looking forward to seeing you again as we defeat this virus and move on with our lives. Take care and have a good virtual school.